All right, today we're going to finish the protein synthesis. Let me just move on here and hit the highlights of yesterday. So DNA, always in the nucleus. The message to get the blueprints out of the nucleus is through messenger RNA. So messenger RNA will be done to remove the message out of the nucleus, and then proteins can be made as the messenger RNA goes to the ribosomes. And then that process is referred to as translation. So we're going from DNA, a nucleic acid, is just written down as an RNA. So we're just transcribing it, basically the same language of nucleic acids. But then we got to translate it into the language of proteins. That's what's going on here. All right, and then we talked about DNA versus RNA and how transcription takes place. And we remove the introns. And now we're looking at messenger RNA and how it codes for proteins. So messenger RNA has to leave the nucleus. It goes to the ribosome, and then proteins are built from it. So how does this happen? How do we make messenger RNA turn into amino acids? Well, basically read as what's called a triplet code. So every 3 letters of DNA makes 3 letters of messenger RNA, which then codes for the various amino acids, which are different colors here. So the ribosome comes in, reads the messenger RNA, reading every 3 letters. And those 3 letters will then say what amino acid goes there at that spot. So TAC transcribes to AUG, and that translates into the amino acid methionine. CGU is arginine, and then GUA is valine, and so on. Every 3 letters is a new amino acid. And that's referred to as a codon. So those 3 letters of messenger RNA is a codon. Codon is a block of 3 messenger RNA bases. And it's read with this chart here called the codon chart. So it doesn't matter what life form you are, every 3 letters will translate into the same amino acid, whether you're a duck or a human or what have you. So if you have UUU as your codon, that is going to become a phenylalanine, or PHE is what it would be there. They usually write it as the first 3 letters. These are written all the same way. The first letter is from the left here. So if you start with a U, you're going to find that box up here. If it's a C, it's going to be down here, then A, and then G. That's all from the first letter. The second letter is coming from the top. So if you're UA, you take the U, you go to the U box here, and then the third letter is over here. So if it's UAU, you just take this U over here and come by here, and you'll see UAU, and will then translate to this amino acid. So, like I said, this is all, doesn't matter what life form you are, whether fruit fly or human, you basically have the same genetic code, the way it's read anyway. The codons all read the same. It is one of those things that support the origin of all life. The code does have duplicates. There are several codons for each amino acid, for most of them, not all. So if you look here at CCU, CCC, CCA, and CCG, they're all code for proline. And if you notice, most of these, the first 2 letters are the same, and that third letter doesn't really matter what it is, it's going to code for the same amino acid. This is a way that we kind of have mutation insurance. So if that third letter is mutated, the DNA is mutated, it really doesn't matter. It's still going to translate into the same amino acid. So while the DNA may be mutated, the protein's still the same. So that's kind of a kind of a way to prevent mutations, at least mutations that could cause a problem. The start codon, all amino acid, or I should say all proteins start with the same amino acid, and that's right here, AUG. So that means all proteins start with the amino acid methionine, MET. So remember, school starts in August, and so do all proteins. So AUG should be your start. If you ever forget how to read this chart, because you might see this on an ACT test, you basically find methionine, and you look at where the A is. Oh, that's right here. The U is here, and then the G is here. So it'll help you try to figure out how to read it. Methionine only has one codon, AUG. All right, there's also some stop codons, as you can see here. You don't need to memorize these ones. The only one you need to know for sure is AUG. The rest of them you don't, because the letters will just tell you to stop when you read the chart. So in the chart, you'll find out when to stop. So the stops are UAA, UAG, and my favorite, UGA, UGA. All right. There's the start, and there are the stops. All right, so how are the codons matched to the amino acids? So we got our first three letters here of DNA. That transcribes. So if you got a T, that becomes an A becomes a U. Remember, this is RNA, so U is there, not a T. T is only in DNA. C's or G's, and so on. So that's how you would transcribe it. There's another RNA that comes in called transfer RNA. And what is it transferring? It's transferring an amino acid. So these tRNAs basically match the messenger RNA. So they're kind of the complementary match. So for every A, this is RNA, so you're going to have a U. The U is going to be an A. The G is going to be a C. So you're going to have these letters that are the opposite complementary side of the RNA, bringing in a new amino acid. Those are referred to as anticodons. Why? Because they're the opposite of the codons. That's simple. So you can see each one is bringing a new amino acid in. Those will be connected to create protein. All right, so the working instructions to make the protein will be on the messenger RNA. The ribosome is going to be the reader of the instructions, the architect, if you will, the builder. And then the person transporting goods to make the protein is going to be the tRNA, the transfer RNA. So the ribosome goes along and reads every 3 bases. And as it reads 3 bases, new tRNA comes in, bringing an amino acid in, which will then attach to the chain as it grows. The next 3 will bring in another tRNA, which brings in another amino acid, and so on. So just keep adding, and we elongate the chain. All right, so as I mentioned, DNA is turned into messenger RNA through transcription. Messenger RNA is changed into protein by the ribosome, and that is referred to as translation. Protein will actually be the actual gene, or what it's going to do, which will then be our traits. And don't forget, tRNA comes in there, bringing the amino acids. All right, so there's 2 basic charts that you could see. This is the square one is generally the one that you will find. It's the one that's on the ACT test. So, if we look at MET, that's always AUG, as you should know. First letter is A here. So we take the A, so we're going to be in this region here if the first letter is A. U is the second letter, so we just see where U and A match up. So we're in this square here. And then G, we just come along here and know it's on the bottom here. So it ends up as MET. Unfortunately, this is a very skill-based activity. And normally when you do a skill-based thing, I show you how to do it. I talk you through it. and then I watch you do it, and by watching you do it, I can usually figure out if you're making a mistake and correct you pretty easy. This generally ends up being a pretty easy assignment. But because I can't watch you actually do it, it might be a little difficult. Another codon chart is this one here, the round one. In this case, you start in the center. So we find A, and we're in this quadrant with the A. Then we go to U, so we're down here. And then we just come along and find G, and then we'll just read methionine. So generally, the first three letters are the ones used here to find the amino acid, the full names of the amino acids. All right, so we could practice here. So you got this template, and you may want to actually do this because you're going to have to do this tomorrow on the breakout room. So just copy down this DNA molecule here, and then transcribe it into messenger RNA. For some reason, a lot of our teachers at this school want you to figure out tRNA. I've never figured out why. No one ever asks you for the tRNA. If you do the tRNA, keep in mind it's the messenger RNA that goes into the codon chart. If you put in the tRNA codes, you'll get something totally wrong. And you'll know it's wrong because your first amino acid will not be MET, methionine. So that should give you a clue you're doing something wrong. All right, so if I were you, I would do the messenger RNA and then plug those in the amino acid chart and see how that works. All right, so when we talk about how cells are organized, the proteins, which are a chain of amino acids made by the ribosomes, the protein factor in the cytoplasm. So that's what the ribosome does, build proteins. Once they're made, they need to be folded correctly. You need to have a good shape for the protein or it doesn't work correctly. So the proteins are folded and twisted into a three-dimensional shape. And that's what happens to the cell. If it's folded incorrectly, it will not work correctly. So that is important that it gets folded correctly. Different shapes will give you different jobs. So like this one here is an enzyme. If that active site isn't correctly made, then it won't work correctly. All right, so the folding basically goes just the amino acid chains referred to as the primary structure, just a line of amino acids. Sometimes they'll start folding with each other, touching each other in two different forms. So the secondary structure will either be this beta pleated sheet, very spread apart, or this alpha helix, which is just a single helical structure. As these fold together, they create kind of this globular structure referred to as the tertiary structure. And if you put several tertiary structures together, you get the quaternary structure. And I think that pretty much sums it up. Basically, tertiary structures like hydrogen bonds. There's a couple of amino acids that have sulfur in them, and those two sulfur amino acids will bind to each other, and that kind of creates these kind of looping situations here. If they're hydrophobic, they want to stay away from water. Cells made mostly of water, so that means it'll try to fold inside itself so that it's not exposed to water, where the hydrophilic amino acids will point outside towards the wall. So remember, proteins run you, and they're made up of enzymes. That's a big function. They're also our structure. So what you see is the structure. As things move and work, that's the chemical reactions of enzymes. So they're very important products used on you. Mutations we'll talk about on another video. That's all for now.